Welcome back, everybody, to another deep dive. I know a lot of you out there struggle with blotchy Western blots.
Oh, yeah.
And today we are going to deep dive into that.
Yes.
We have a great article here from Assay Genie that dives into the causes and solutions.
Okay.
So,
Blotchy Western blots, they can be a real pain.
oh, for sure.
Whether you're just starting out or you've been doing this for years, they never seem to go away.
Right.
Yeah, and Western blots are so fundamental, They're everywhere, like all over in disease research, drug development, all that.
You think you have it down, and then
Yes, exactly.
you get those blotches, and it's like, oh no, what
They
happened?
just completely mess everything up.
Yeah, we're talking uneven bands, streaks, patches,
Yeah, it's like where there should be a clear signal, it's just blurry and messed up.
You can kind of see what's going on, but not really.
Yeah, it's like trying to read a faded blueprint.
Exactly.
But the problem is, Western blots aren't just about getting pretty pictures.
No.
We need to be able to really see how much protein is present.
Yeah, you need those clear results.
Yeah, to be able to quantify things, And if we can't trust the data because it's all blotchy, then what are we even doing?
It can totally throw off your whole experiment.
And lead to the wrong conclusion.
Exactly.
So it's not just about how it looks.
Right.
It's really about getting good data.
It's about the integrity of the whole thing.
Exactly.
So this article we're looking at, it lists out a bunch of the most common causes.
Okay, yeah.
One that really stood out to me is incomplete protein transfer.
Oh, yeah. I've been there.
It's like you're moving and only half your stuff gets to the new place.
That's a great analogy. And it all comes down to how well the proteins bind to the membrane during the transfer.
So you really need those proteins to latch on tight.
Oh, yeah. Otherwise, they just get left behind.
So it's more than just physically moving them.
Absolutely,
You need the right conditions for them to bind.
For sure.
Okay, what about uneven sample loading?
Oh, that's another big one.
I feel like that's such an easy mistake to make.
It really is. And if you don't put the same amount of protein in each well, you're going to have problems.
Totally. Like, how are you even supposed to compare then?
Yeah, it's like comparing apples and oranges. You need to start with a consistent amount.
Makes sense.
You need that baseline.
Okay, and then there's gel quality too.
Oh, don't even get me started on bad gels.
One time I poured this gel and it looked perfect, but when I ran my samples, it was a disaster.
That's the worst.
The bands were all over the place.
Ugh, it's so frustrating when that happens.
I know.
Because the gel is like the foundation of the whole thing.
Right.
And if it's not right, then nothing else matters.
It's like building a house on sand.
Exactly. It's just not going to work.
So how do you even avoid that?
You got to be really careful with the recipe,
Yeah.
making sure everything is measured out exactly right.
Your percentages and all that.
Yep, all of it. Even the temperature can mess things up.
Boy.
And those little air bubbles, those can be a nightmare too.
Oh, yeah, those can cause streaks and blotches, right?
They sure can.
Okay, so we've talked about a few things that can cause blotches.
Yeah, but there are ways to fix them too.
That's what we're going to deep dive into next time.
I can't wait.
Me too. We'll talk about how to actually solve these problems and get those nice, clean bands.
Sounds good.
So stick with us.
We'll be right back.
Don't go anywhere. Okay, so we've talked about some of the things that cause those blotchy Western blots.
Yeah, a lot of things can go wrong.
It can feel overwhelming.
It really can.
Yeah.
But don't worry.
Yeah.
We're going to figure this out.
Okay, so where do we even start?
Well, the key is to be systematic about it.
Okay.
So instead of trying to fix everything at once, you really got to take a step back
Okay.
and look at the overall pattern of the blotches.
Like, what do you mean?
Like, where are they? Are they all over just in certain areas?
Oh, okay.
They can give you some clues about where the problem might be.
Oh.
Yeah, it's like detective work, gathering evidence
That makes sense.
before you start making any conclusions.
Okay, so let's break it down step by step.
Okay.
Let's start with sample preparation.
Okay.
What do we need to keep in mind when we're loading those protein samples?
You have to be so precise when you're loading those samples.
Okay.
Inconsistent loading is one of the biggest culprits for blotchy bands.
Like even just a tiny bit off.
Yeah, even a little bit can make a difference.
Wow.
It's like baking a cake. If you don't measure everything out exactly right,
Right.
it's not going to turn out how you want it to.
So use a calibrated pipette?
Yes, definitely. And take your time.
Okay.
Make sure each well gets the same volume.
Okay, got it.
But even if your pipetting is perfect,
Yeah.
you could still have problems if your samples have different amounts of total protein.
Oh, right. Of course.
That's where loading controls come in.
Loading controls.
Yeah, they're basically proteins that are expressed at pretty consistent levels
Okay.
across all your samples. So if you include a loading control on your blot, you'll have a reference point.
To compare to.
Exactly. So if your loading control looks good,
Okay.
but your target protein is all blotchy,
Yeah.
then you know the problem isn't the loading.
Interesting.
It's something else further down the line.
Okay, that's a good tip.
Yeah, loading controls can be really helpful.
Okay, what about the gel itself?
Ah, the gel.
Yeah, you were saying earlier how it's like the foundation.
It really is.
So how do we make sure it's a good foundation?
First of all, you got to follow that recipe exactly.
Okay.
Especially when it comes to the percentages of acrylamide and bisacrylamide.
Okay.
And the catalyst and the temperature. Everything has to be just right.
Wow, okay.
Even a tiny change can affect the pore size
Okay.
and the
Yeah.
structure of the gel.
And then that messes up how the proteins move.
Exactly. It can totally disrupt the migration.
Okay, so recipe is key.
Yes. And don't forget to degas the solution.
Gas.
Yeah. You know those little air bubbles? They can get trapped in the gel.
And cause problems.
Oh, yeah. They can create streaks and blotches.
So how do you degas it?
You just put it under a vacuum for a few minutes before you pour the gel.
Oh, okay.
That'll get rid of most of the bubbles.
Cool. Good tip.
It's all about the details, right?
Right. The little things matter.
They really do.
Okay, so we've got our perfectly poured gel.
Awesome.
Now we need to transfer those proteins onto the membrane.
The transfer, yes.
How do we avoid those incomplete transfers?
So the transfer is all about creating the right sandwich.
Sandwich?
Yeah, with the gel and the membrane,
Right.
they have to be in close contact with no air bubbles in between.
Okay.
That way the proteins can transfer evenly.
So no wrinkles or anything.
Exactly. You want it nice and smooth.
Okay, so the sandwich setup is important.
Very important.
What about the transfer buffer?
Oh, the buffer is crucial too.
Okay.
You got to use fresh buffer every time.
Every time.
Yeah, over time it can get depleted
So,
or contaminated, and that'll mess up the transfer.
so just like with cooking, you need fresh ingredients.
Exactly. Fresh buffer for the best results.
All right, what about blocking and antibody incubation?
Okay, so blocking is all about preventing non-specific binding.
Okay.
And there are a lot of different blocking agents out there.
Like what?
Milk's a common one.
Oh, like regular milk?
Yeah, or bovine serum albumin or casein.
Okay. I've heard milk can sometimes mess with certain antibodies.
Yeah, that can happen.
So how do you know which one to use?
Honestly, it often comes down to trial and error.
Oh,
You can start with a standard solution,
like what?
5% milk in TBST,
Oh, okay.
and see how it goes. If you get a lot of background noise,
Yeah.
you can try a different blocking agent
Okay.
or adjust the blocking time or temperature.
So it's all about finding what works best for your specific experiment.
Exactly. It's not a one-size-fits-all kind of thing.
And then antibody incubation, that's another big one, right?
It is, yeah. Antibody optimization can be tricky. The concentration of your antibodies, the incubation time, even the buffer you use, all of that can affect your results.
I spent weeks optimizing antibodies for one experiment.
Oh, yeah, I've been there.
It was like a puzzle.
It really can be.
But that's why it's so important to be systematic.
Absolutely. You got to change one thing at a time and see what happens.
Okay, and what about washing?
Washing is so important.
Okay.
You got to get rid of all those unbound antibodies.
Otherwise.
You'll get background noise, and that'll contribute to the blotchiness.
So how do you make sure you're washing enough?
Use plenty of wash buffer
Okay.
and agitate the membrane gently.
Like on a shaker or something.
Yeah, and stick to the recommended number of washes.
Okay, got it.
It's all about being thorough,
And then finally, signal detection.
right? The moment of truth.
Are there any common mistakes there?
Consistency is key.
Okay.
Whether you're using chemiluminescence or a chromogenic substrate, make sure you're applying it evenly. And using the same exposure time for all your blots.
Got it.
That way you can compare them accurately.
Makes sense. Wow. It's a lot to think about.
There is, but it's not rocket science.
Yeah.
Once you break it down into steps, it's manageable.
I'm feeling more confident already.
Great to hear.
Okay, so we've covered a lot of ground here.
We have.
But there's one more thing we need to talk about.
Oh, right.
Documentation.
That's so important.
We'll dive into that next time.
Good.
All right, so we're back. And we've been talking all about those pesky, blotchy Western blots.
Try to get rid of them.
Yeah, figuring out what causes them, how to troubleshoot.
It can be a real mystery sometimes.
But we've covered the technical stuff, sample prep, antibodies, all that.
Yep.
But there's one more thing we got to talk about.
Oh, yeah.
Documentation.
It's so important, and people don't always realize it.
I know. It's easy to forget in the heat of the moment.
It really is. You're focused on the experiment.
Yeah, you're just trying to get it done.
But trust me,
Yeah.
taking good notes is crucial, especially when it comes to troubleshooting.
So, like, what kind of notes are we talking about?
Everything.
Everything.
Well, okay, maybe not everything, but
Yeah.
all the key parameters of your Western blot.
Okay.
Gel recipe, transfer conditions, blocking agent, antibody concentrations, incubation times, washing steps, all of it.
So it's not just like writing down the protocol.
No, it's more than that.
Okay.
It's about recording exactly what you did for that specific experiment.
Oh.
And any deviations from the protocol.
Variations.
Yeah, like, did you increase the blocking time? Did you try a different antibody dilution? All that stuff.
Oh, okay.
Like taking notes while you're reading a textbook.
Right.
Helps you remember the important stuff.
And then you can go back and reference it.
Exactly.
So when you get those blotchy blots,
Yep.
those notes become your best friend.
They really do. You can compare different experiments.
Okay.
Look for patterns.
Like what?
Maybe you always get blotches when you use a certain batch of transfer buffer.
Interesting.
Or when the temperature in the lab is too high, you know.
So it's all about data collection.
Yeah, even when things go wrong.
Even when you get those blotches.
Those blotches are telling you something.
They are.
It's just a matter of figuring out what.
It's like they're trying to communicate with us.
Exactly.
And good documentation helps us understand.
It's like a scientific journal, a record of your journey.
I like that. A Western blot journey.
And those records are also important for reproducibility.
Oh, right. Of course.
If someone else wants to repeat your experiment,
Yeah.
they need to know exactly what you did.
So it's not just for us. It's
Well,
for the whole scientific community.
It's about sharing knowledge.
Making sure everyone's on the same page.
And who knows, maybe your notes will spark a new idea for someone else.
Yeah, that'd be cool.
It's all connected, you know.
So those blotches, those little frustrations,
Mm,
they can actually contribute to something bigger.
they can.
That's pretty amazing.
Science is full of surprises, right?
That's why we do it.
Exactly.
Well, I think we've covered just about everything there is to know about blotchy Western blots.
Yeah, I think we've pretty much cracked the case.
We've got the causes, the solutions, the importance of documentation.
We're ready to go out there and fight those blotches
Armed with knowledge.
and good note-taking skills.
So to all our listeners out there,
Yep.
don't be afraid of those blotches.
Embrace them
Learn from them.
and keep those lab notebooks handy.
And remember, you're not alone in this.
We're all in this together.
So keep experimenting.
Keep learning.
And keep those blots clean.
The best we can anyway.
That's right.
Thanks for joining us for another deep dive.
We'll catch you next time.